In 1954, looking through a phase contrast microscope at a sliver of rabbit muscle, two separate teams of scientists noticed something strange. As the muscle fiber contracted, certain bands within it, the dark A bands, didn't actually get shorter. Instead, the lighter bands on either side seemed to slide inwards, disappearing into the dark ones. This wasn't a story of proteins shrinking. It was a story of them sliding past one another. This single observation, the sliding filament theory, fundamentally changed our understanding of movement itself. The engine driving this sliding is a protein named myosin 2. It doesn't just sit there, it's a true nanoscale motor. Imagine a molecule with two heads attached to a long winding tail. These heads are the active part, the part that binds, pulls, and releases. Each head is a complex machine that converts chemical fuel, a molecule called ATP, into pure mechanical force. There are billions of these tiny engines packed into every single one of your muscle fibers, waiting for the signal to pull. The process is a meticulous four-step cycle. First, a molecule of ATP binds to the myosin head, causing it to detach from the actin filament it was holding onto. Second, the myosin head breaks down the ATP into ADP and a phosphate, using the energy released to cock itself into a high-energy position, like pulling back the string on a bow. When it rebinds to the actin filament, it releases the phosphate, triggering the power stroke, a tiny, forceful tug. Finally, the ADP is released, and the head is ready for a new ATP molecule to start the cycle all over again. But why aren't your muscles constantly contracting? This incredible machinery needs a switch. That switch is controlled by two other proteins, tropomyosin and troponin, which act like a safety lock. Tropomyosin is a long filament that snakes around the actin, physically blocking the myosin heads from binding. When a nerve impulse arrives, it triggers the release of calcium ions. These ions are the key. They bind to troponin, causing it to change shape and pull the tropomyosin wire out of the way, exposing the binding sites. Suddenly, the engines can connect with the track. How much force does one of these power strokes generate? The numbers are almost comically small, measured in piconewtons. A single piconewton is the force required to break a single hydrogen bond. One myosin motor produces maybe 5 to 7 piconewtons of force. It's a whisper of a push. But your strength isn't about one motor. It's about the near-simultaneous, coordinated action of trillions of them. The ability to lift your phone is an emergent property of piconewtons adding up in a profoundly massive way. So, what are the limits? If you just activate more muscle fibers, can your strength increase indefinitely? The answer is no, and the reason is fascinating. The primary limitation isn't your muscles, it's your brain. Your central nervous system acts as a governor, a safety inhibitor that prevents you from activating all of your motor units at once. It does this to protect you. A full, unrestrained contraction could generate enough force to tear tendons from your bones or even snap the bones themselves. Your perceived limit is actually a carefully calculated safety margin. This neural inhibition is what makes stories of hysterical strength so compelling. In moments of extreme duress, life or death situation, the brain's inhibitory signals can be overridden. The flood of adrenaline and other hormones can temporarily remove the safety locks, allowing a person to access a far greater percentage of their muscular potential. This isn't supernatural. It's a glimpse of the raw physical power your body carefully prevents you from using every day. The hardware has always been capable of more. The software simply forbids it. Even at the molecular level, not all myosin is created equal. You have different versions or isoforms of this motor protein. These define your muscle fiber types. Fast-twitch fibers contain a myosin isoform that burns through ATP incredibly quickly, allowing for rapid, powerful contractions perfect for sprinting or lifting heavy weights. Slow-twitch fibers have a more efficient myosin that cycles slower, resisting fatigue and making it ideal for endurance activities like running a marathon. Your personal ratio of these fibers is largely determined by your genetics, a blueprint for your athletic potential. This brings us to a final, profound question. If the engine of movement is just a protein coded by our DNA, could we change it? Could we design a better myosin? Bioengineers are already exploring how to create synthetic molecular motors. The idea of editing the human genome to produce myosin isoforms with higher force output or greater efficiency is no longer pure science fiction. It forces us to wonder, if we can rewrite the fundamental protein of power, what does strength even mean? Is it the force you're born with, or the force you can design?
Monotony has grown fast. Nearly 2,700 people have watched in the last few weeks, yet less than 0.03% are subscribed. It would mean a lot if you joined by hitting subscribe. And again, thanks for watching.